Hello, welcome to Biochemistry Simplified. We are here again. We're looking at uh, biostatistics for biological sciences. We started this some time ago, born out of curiosity and questions. Um, Biological scientists and students, postgraduates, graduate students are asking to know exactly what type of statistical analysis that will be required for specific biomedical and biology researches. So we've looked at the basic analysis of data in biological sciences, classified into basically qualitative and quantitative analysis. Okay, so we've also looked at a different type of assay, that bioassay that we can examine in biological sciences in the previous lectures. So I'll employ you to look through those lectures. We just want to move right along, and today we'll be looking at the levels of measurements in biostatistics. So when you now know the type of research you want to carry out, if it's a type of analysis you like to apply to your research, you want to now know the levels of measurements. This is very key. So today we'll begin by looking at the levels of measurements. Levels of measurements. Okay, so in biostatistics and research, understanding the levels of measurement is very essential because they help you to determine the appropriate ways to organize specifically, and then to analyze and interpret data. Those are very key things in which the levels of measurement is of great and huge importance. Okay, so they help you to organize. So you want to know what's the level of measurement, of what importance it is. So I just like to put that down. The level of measurements, the level of measurements, level of measurement is essential because is essential because of these three meetings. Number one, or you say A, number one is essential because they help you to determine, help to determine they help to determine the appropriate ways to organize, analyze, and interpret data. So that's actually the three key things. The levels of measurement does these three things for you. They help you determine how to a to organize, all right. Then you have to analyze, and the third one, to interpret the data. To interpret data. Yes, this is very key. So when you want to now move into your research proper, you have to look at all of this. How do I organize whatever it is that will be the output or outcome of my research data? How do I analyze it? And most importantly, how do I interpret it? Because if you are unable to interpret the data, the research is as good as useless, because it's your interpretation of your scientific results and data that you have organized and analyzed in such a way that gives meaning to the entire research. Okay, so there are four primary levels of measurements. There are four primary levels of measurements, which includes the nominal, ordinal, interval, and ratio levels of measurements. So we quickly put that down. There are four There are or primary, four primary levels of measurements. Number one is the nominal level of measurements. The nominal level of measurements. Then you have you have the ordinal level of measurements. Ordinal level of measurements. We have three as the interval level, interval level of measurements, and of course the last but not the least is the ratio level of measurements. The ratio level of measurements. These are the four primary levels of measurements, and each of these levels of measurements, they offer different kind of information about the data and the impact, type of statistical analysis that should be applied. So the statistical analysis that will be applied for nominal level of measurement is going to be quite different for the statistical analysis that should be applied to a ratio level of measurement. Okay, so the keynote is that each of these levels offers different information about the data and how it impacts the types of statistical um, analysis that should be applied. So each of these it differs. Each of these differs in their application, in their application, in their application to statistical analysis. In their application to statistical analysis. Okay, so we'll begin to look at each one of them: the nominal level of measurement, the ordinal level of measurement, interval level of measurement, and of course the ratio level of measurement in the next page. Okay, so we'll begin with the first one, which is the nominal level of measurement. The nominal level of measurement. All right. So what is it about? The nominal level of measurement is the most basic, and it classifies data into distinct categories. So this level of measurement, number one, is key point that is the most basic. Okay, most basic level of measurement, and it classifies data into distinct categories. Most basic and classifies data. It classifies data into distinct categories. Classifies data into distinct categories. Into distinct categories. That's for the nominal level of measurements. Okay, so when they do this classification, they do not have any inherent order or ranking. So in nominal data, you do not have no inherent, okay, no inherent order or ranking. No inherent order or ranking. Just like we saw when we were looking at um, qualitative and quantitative analysis, where we also make mention of some of these levels: nominal, ordinal, and the rest of it. Okay, so they do not have any inherent order or ranking because they are basically qualitative or categorical in nature. That is, They categorizes and they represent names, labels, or groups without numerical meaning. That means they do not have numerical value attached to it. They are majorly categorical or qualitative in application. Okay, so they are basically categorical or qualitative in application. Okay, so I write that down. They are qualitative. They are qualitative or categorical in application. In application, okay, as they represent no labels, groups without any numerical meaning. Okay, as they represent groups, alright, um, or labels, like general labeling without any numerical meaning. That means they do not have specific numbers. All right. So from what we see with nominal data, they have these key features or characteristics. For nominal data, we have features or characteristics of nominal data. That means when you see an analysis, how to identify that is nominal data, these are the things that you begin to look at to identify that is the nominal level of measurement was applied. So when you do that, you begin to see things like for the features, you see that there's no specific order, okay, no order or ranking. So categories cannot be ranked or ordered. Okay, so no specific order. Then you begin to see no arithmetic or mathematical, sorry about that, no arithmetic operations, no arithmetic or mathematical operations. That is another key feature or characteristics. That is, since they do not have um, order or ranking, they are just labels. They do not have mathematical calculations that can be performed. So this can also imply no mathematical calculations are carried out or performed. No mathematical calculations are applied. Oh, sorry about this. All right. So no mathematical calculations are applied. So that's one, two major characteristics. There's one more. You have mutually exclusive. They are mutually exclusive. That means each value belongs only to one category and categories are distinct from one another. So mutually they are mutually exclusive. Okay, they are mutually exclusive. That means um, categories are distinct from one another. Okay, categories or labels are distinct or different. Okay, They are distinct or different from one another. From one another. So each value belongs to each category specifically. Examples of this kind of nominal data includes species of animals, blood groups, eye color. Okay, so for this type of nominal level of measurement, examples include Examples include you have species of animals, e.g., lions, alright, tigers, bears. Okay, so these are like labels. Alright, so you have species of animals, then you also have blood type as another example. Blood types, where you have species of animal, you could have things like dogs, rabbits, all right, like categories: lions, okay, tigers, etc. Then blood types, you could have things like blood group A, blood group B, blood group AB, or blood group O. Then you also have examples, things like your eye color, like phenotypic expressions, where you begin to see a group of categorization, such as having blue eyes, brown eyes, or green eyes. Okay, blue, green, or brown. All right, so this type of level of research is very important and very useful for classifying biological entities into categories, like we have emphasized before. So, a clear example is that scientists might categorize animals in a study by species or classify people based on their blood group or blood type, just to study a health trend. Okay, so that category is there. They are just looking at the categorization, all those with blood group O, all those with blood group B or AB. There are no specifics, just to have these people to study a type of health trends. So when analyzing nominal data, researchers often use things like statistical tools that can be used during a nominal level of measurement is that they use research tools such as frequency counts, all right, research tools such as frequency counts and present data in tables or pie charts. So statistical tools that can be used, don't know if this space down can do it. So statistical tools can include, sorry, tools can include pie charts, pie charts, and frequency counts. What else? We have pie charts. We can have a data presentation in tables, tabular, tabular data, etc. We also have things like your chi-square, right? Your chi-square. Okay, so that's basically for nominal data. We'll now move on to the ordinal data. Okay, so next slide. So the second category that we'll be looking at is what? Your ordinal data. We just concluded looking at nominal level of measurement, and now we are looking at the ordinal level of measurements. Great. So the ordinal level of measurement uh, is the type of measurement that organizes data into categories that have meaningful order. For the nominal, remember that we said no order, no inherent ranking. While for ordinal, now you have um, the level of measurement organizes the data into categories that have a meaningful order or ranking. However, the intervals between ranks are not equal, and specific differences between ranks are what unknown. Okay, so they don't know specific differences, but there is a ranking and there is a meaningful order. Very important, as different from nominal level of measurements. So we just quickly note that down. The ordinal level of measurement is said organizes it organizes data into categories, organizes data into categories and that have a meaningful order or ranking. That's very key. So the key word here is order or ranking. Meaningful order or ranking. Even though the specific difference between each order are not known, but there is a meaningful order or ranking. Features with which you know um, that a ordinal level of measurement is applied includes so features or characteristics characteristics of ordinal level of measurement, ordinal level of measurement includes so you have these features, sorry, so ordinal level of measurement, so you have these features here. Let me try to draw a straight line now. Okay, that's great. So the first feature, like I noted, is that there is what? An order of categories. Order of categories. Alright, this is very key. Categories have a logical sequence or ranking. Then the next clear feature is that you do not have an they have an unequal interval. Alright, the difference between them are not known. So an unequal what? Interval. That's the space between each category is not necessarily uniform or measurable. Then the third and key feature with which you can categorize the level of measurement is that they are comparative only. Values are relatively positioned when not when there are no precise differences. So they are for comparative analysis only. Okay, they are comparative only. Okay, so examples of researches, biological research that could and analysis that could take on such ordinal level of measurements includes examples include things like disease severity. Okay, I can take examples of this ordinal level of measurement. Examples include things like your disease severity. All right. So in this, you do not have specific number, but you could tell, for example, that maybe if you look at say temperature, you could tell if it is mild, if it's severe, even maybe a pain. Pain is mild, is severe, or is moderate. Okay, or is mild, moderate, or severe. Okay, so that's one key example that you can look at, and you know that the ordinal level of measurement is being applied. The other one is socioeconomic status. For instance, you are carrying out certain research analysis, and socioeconomic status can also come into play, where you have things like the low, the middle, high class, low class, middle class. All right. So socioeconomic status is another key point, where you have uh, low low income economic countries, or um, middle income, or high. Okay, like a classification, like a class. All right, so you have that example of socioeconomic, and the other one is like a pain scale, which is almost very, if not directly related to disease severity, where you have like a pain scale that also shows mild, moderate, or severe, where you have like a scale where one implies that it's mild, and you have two implying that it's moderate, and three implying that it's severe. Okay, so you have this pain. One, uh, the scale, one implies that it's a mild pain, two implies that it's a moderate pain, and uh, three three implies that is what a three implies that is a severe pain. So that's basically the example. Ordinal data, they are often used in medical research, in biological research, in assessing ranked outcomes like patients' pain levels or stages of diseases. Although we know the order, e.g., severe pain is worse than mild, the exact difference between categories isn't what's measurable. Okay, the statistical test that can be used. So in research, this is actually used in is kind of used mostly in medical, or rather used. Sorry about that. Oh dear, sorry. So it's used in biological or medical research, basically. All right, like I said, it's used in biological medical research, and it's used to assess ranked outcomes. Okay, like patients, pain levels, or stages of disease. All right, so medical research to um, assess ranked outcomes. To assess ranked outcomes. outcomes, okay, patients, such as patient's pain level, patient's pain levels, or stages of disease. Pain levels or stages of disease. This is very, very key. Then statistical tools that can be used, or tests that can be carried out for ordinal level of measurements include, okay, statistical tests or tools that can be used in carrying out um, this type of ordinal level of measurements include things like your U test, U test, then you have Manny Whitney. Okay, this actually Manny Whitney you test and your okay, I take that again. So you have statistical test, you have Man Whitney, Man Whitney you test. Then you also have Crystal Wallis test, Crystal Wallis test, Crystal Wallis, Crystal Wallis test. Okay, and this test analyzed um, ordinal data, while virtual representations like bar charts can show their distributions. So these two tests they organizes, all right, or analyzes. It analyzes ordinal data at the ordinal level of measurements, and that's basically it. And bar charts, bar charts shows um, distribution of visual representation. So you have also bar charts that shows distribution, the distribution of what? Of visual representation, of visual representation of the data. Great, that's for your ordinal level of measurements. Okay, then the next one we'll be looking at is the third one on that list, which is your interval level of measurement. These levels of measurements they are key because when you know the levels of measurements, you will know exactly how to go about your sample collection, having figured out the type of major analysis you'll be carrying out, whether